First, it's really an honor to be part of welcoming President Rodriguez to the School of Public Health. And I wanted to tell you just a little bit briefly today about a relatively new program in climate change and emerging infectious diseases that actually grew out of another successful graduate training program that's been more longstanding in the Department of Biomedical Sciences in biodefense and emerging infectious diseases. And the founding faculty in this CCEID program, which is what we tend to call it, are split evenly between the Biomedical Sciences Department and the Department of Atmospheric and Environmental Sciences. And as all of the biomedical sciences faculty have Wadsworth-based research programs and also interact closely with our DOH colleagues at the Bureau of Communicable Disease Control. We are also very excited to recently become part of a CDC Center of Excellence in Vector Biology in the Northeast region that's coming out of Cornell. And we are also very much looking forward to the university's pending Institute for Climate and Health. And so, therefore, this is an extremely collaborative and transdisciplinary program, and this is probably the most important slide that I'll share on this today. And so as far as faculty and expertise, Laura Kramer is a research scientist who studies mosquito-borne viruses such as West Nile virus and dengue and Zika, and whereas Jan Kahn studies mosquitoes that transmit malaria. And on the climate side, we have Oliver Ellison-Tim and Matthias Wee, and both of them study climate variability and climate change, and Chris Thorncroft co-leads the program with me, and when he's not thinking about monsoons in West Africa, he is worrying about keeping and getting the New York State Mesonet up and running, and that's a very elaborate and dense series of weather stations throughout the entire state of New York. I should mention that virtually everybody on in this group, with the exception of myself, has very extensive international programs as well as local programs. And so we have a joint seminar program that we also do jointly with other folks from the School of Public Health in the Environmental Health and Science program, and we're doing some cross-teaching. But from the research side of things, the group is really interested in particularly how does climate change affect the future of infectious disease incidence, with, of course, the intent that if we can better predict, then perhaps we can also intervene and prevent some future epidemics. And an important goal is to be able to do this at a very mechanistic level with respect to very specific climate variables that affect specific biological aspects of transmission. And one of the ongoing research programs or questions is on West Nile virus and how climate may have played a role in it distributing across New York State after its introduction into New York City in 1999. And as many of you know, West Nile virus is transmitted by mosquitoes, and it circulates within bird populations that serve to amplify the virus. And we only have a problem when we get in the way of this transmission cycle and become the next meal for one of these infected mosquitoes. On a very, very superficial and generic graphic here showing some of the basics of vector-based transmission. What we know from other studies is that each aspect of a cycle can be affected by multiple different climate variables. And I should also note that that from the perspective of viruses like West Nile virus and other arboviruses, this link doesn't happen. You actually need to have a vector to get the transmission. But for other diseases, like, for example, Yersinia pestis and plague, one can also have an overflow here. But the main point I want to make is the vector-based transmission here and the different climate variables that can affect the vector in terms of survival of mosquitoes or fleas or ticks, temperature, humidity, rainfall. Rainfall can affect the population density of the hosts, particularly because of food availability. Most pathogens, be they viruses, bacteria, or parasites, are affected by temperature as far as their development in their growth rates, and then also the likelihood or the extent of things that hosts like people who are not part of a natural wild transmission cycle getting involved and becoming infected with some of these diseases can be very, very affected by landscape or forest ecology issues. This is largely because when people start encroaching on territory that has been where the animals are in the forests and living like in suburbs and at the edge of deforested areas, puts people more in contact with those natural transmission cycles. So I'm not really going to go through data. I just wanted to show you two slides that show a little bit of the kinds of ways that we can look at this. And this is this is just a slide that's showing the incidence of West Nile virus in mosquito pools that were collected during surveillance across New York State, and I'm showing just from Westchester County. And so and this shows since 2001 up to 2014. All the red guys are West Nile virus positive mosquito pools, and this is just showing the summer months for each of the years. Just two different ways of showing it, and you can see the high points when there are peaks of transmission. This can be compared and overlaid with the temperatures, and this is just showing the maximum average temperatures for the different seasons. So we can look for correlations that way, and also we can add in additional factors such as humidity by including a drought index, and then in this case the incidence of West Nile virus for each of those summers is shown by the size of one of these bubbles, and the position on this grid lets us correlate the temperature and the wetness, so that we can again look for correlations that way. And so this is ongoing, and as the project develops, the tools will all be applicable, certainly to apply to the greater Northeast region, as well as additional mosquito-borne pathogens and also other arthropod vectors. And I just wanted to acknowledge the sources of the climate data. Also, as far as Brian Backinson at the BCDC is invaluable for providing all the mosquito trap pools and so on. Two students have been involved in the project. Kate is a graduate student in Jan Kahn's lab, and Sarah Quinones is an undergraduate who worked with on the climate side with Oliver, and certainly we've had some really important funding that I'd like to acknowledge. And so with that, thank you.